Hi, everybody. Welcome to the World Stem Cell Summit session by the Cord Blood Association. I hope you're all geared up to hear some exciting news about advances in the cord blood field. And over the next hour, I and my co-panelists will be addressing some of the questions that are facing the cord blood industry today, and we'll all share our expertise. We're all going to introduce ourselves. I'll go first. I'm Joanne Kurtzberg. I'm a pediatric hematologist oncologist at Duke University Medical Center. And I also am a hematopoietic stem cell transplanter and a cord blood transplanter. And in fact was involved with the very first cord blood transplant, which was in 1988, which is now 35 years ago. Which is hard to believe. And I can say that that patient is alive and well, fully engrafted with his baby sister's cord blood, which he received when he was 5 years old. So we know cord blood can be a durable source of hematopoietic reconstitution after myeloablative chemotherapy. I also was involved in doing the first unrelated cord blood transplant in the world at Duke in 1993 and starting a public cord blood bank called the Carolinas Cord Blood Bank at Duke in 1997. And our bank subsequently became FDA licensed after FDA issued guidance for licensure of public banks first in 2011 and finalized in 2014. Our bank received its license in 2012, and you'll hear during this session why we think cord blood licensure is important and also has added quality to the manufacturing field, not only of cord blood, but of derivative products. In my work today, I still perform cord blood transplants, but and in that work have focused on using cord blood as a donor source for kids with leukodystrophies and inherited metabolic diseases. And now I'm using cord blood as a therapy that's infused without any preparative regimen. So just as a smart drug, if you want to think of it that way, to treat children with various types of acquired brain injuries. Finally, I run a GMP lab that manufactures cord tissue, mesenchymal stromal cells from third-party donors, and we have those cells in clinical trials in children with autism, children with cerebral palsy, babies with hypoxic ischemic encephalopathy, and other conditions. So I've sort of run the gamut of cord blood as a source of hematopoietic stem cells, cord blood for banking, and then cord blood as a starting material for manufacturing other types of therapeutic cells, which I think personally will be part of the future of cord blood and birthing tissues over the next decade. So next, I'll ask Colleen Delaney to introduce herself.
Thanks, Joanne, and thanks to the World Stem Cell Summit for inviting us to have this discussion with you guys. My name is Colleen Delaney. I too am a stem cell transplant physician by training and started that training now over 20 years ago and at the Fred Hutch in Seattle. And it was really during that time that I fell in love with bone marrow transplant and as a form of immunotherapy, and also recognized that there was a huge need to improve the outcomes for patients undergoing stem cell transplant, especially those who didn't have a donor. And so I too have spent essentially my whole career working in the field of cord blood transplant, but also much to my surprise, started developing research therapeutics and running a lab at the Fred Hutch for many years, and effectively developing a platform that was for stem cell expansion, in particular, initially around doing patient-specific expanded products in the setting of cord blood transplant. And we started this work a long time ago, and really saw the full potential of what one could do with cord blood starting material. So from that time, I spent my career then developing these therapies, initially, as I said, expanding stem cells from cord blood to give to patients undergoing cord blood transplant in a patient-specific fashion, but then moved to allogeneic and off-the-shelf cell therapies. And then even further develop this, as Joanne was saying, cord blood is not just a bag of stem cells, but we also now, in my career now, I have started a company. I'm the founder and chief scientific officer at a company called Devera Therapeutics, where we have taken this technology that I originally developed at the Hutch into the company, and we are focused on the development of allogeneic and off-the-shelf cellular immunotherapies that include expanded progenitor cells, but not for cord blood transplant anymore, but expanded progenitor cells and natural killer cells, as well as macrophages, all derived from cord blood. So I think from this, I'm going to approach this conversation from that lens. I still am a transplant physician. I still see patients. So I definitely wear that hat of the need for cord blood in the area of stem cell transplantation. But now I also work with Devera in developing these cell therapeutics. And so I think as we, talk today, I'm going to be coming at it from cord blood as a cellular starting material and what we think about as a small biotech startup in working with banks and other physicians to develop these products for therapeutic intent.
Great. Okay, our next participant is Marcy Finney from the Cleveland Cord Blood Bank.
Hi, I'm Marcy Finney. I'm the executive director of the Cleveland Cord Blood Center, which is a public cord blood bank located in Cleveland, Ohio, which was established in 2008. So when we started the cord blood bank, it was still, cord blood use for transplant was still on its way up. And what I think where we have some influence is that we realized in the middle of our growth phase that just focusing on using cord blood for transplant was probably not going to be a sustainable business model. So my background is I started in cord blood. I have a chemistry degree, and I was started in basic science research with our founder and medical director, Mary Laughlin, who worked with Joanne Kurtzberg at Duke. And we were doing basic science research, and then also at that time realized that there was no cord blood bank in the Midwest. There was Duke, and there was New York, and there was St. Louis. So we've got some family funding to start this cord blood bank. And then as I progressed in the bank, I also was able to get my business degree. So I have a little different hat than the rest of the panel, that I'm not a clinician, but I have a science background. And I think our bank realized that we need to find other uses for cord blood in order to make the banking model sustainable. So we always have our eye on the right cord blood units for transplant, but we've also worked on utilization and how can we influence that from a bank perspective by doing research and development, and also to support biotechs like Colleen's in providing cord blood units to them for use so that we can keep the industry alive. Cord blood's such a unique industry. I had the pleasure of meeting 2 cord blood transplant survivors early when I was a research technician, and their stories have stayed with me through my whole career and make me really dedicated to knowing that there are people out there that this is obviously often the only option for them. One was Nathan Mumford, who was an African-American man who had had cancer early in his lifetime. He needed a stem cell transplant, and he literally found one cord blood unit that was a match for him. So without that one unit, he lived a really full life. I got to see him get married, and he really appreciated that opportunity. And another patient, Diana, who was a Caucasian woman, got cancer late in her life, and also could not find any matches. I mean, she was in dire straits planning her funeral, and she refers to herself as the ultimate recycling project, trash to treasure. Cord blood saved her life, and it was something that's normally thrown away. So I think it also, I'm really keen to think about the donor perspective, because that's where I started in cord blood banking, was talking to donors. We have collection sites in Cleveland, Atlanta, and San Francisco across the country. So I've gotten the chance to talk to many different donors. And their perspective is really different depending on their background, depending on what they know about science. So I think the donor piece is also really important, and it's, they're giving an altruistic donation and their intent is to help someone else. So I think that line that goes through cord blood banking really is something that motivates me. So I'm really happy to be part of the panel today.
Thank you, Marcy. And last but not least is Ramit Simantov, who is from GametaCell and will tell us about the amazing advances they're making with cord blood.
Thank you, Joanne, and thanks so much for the invitation to participate in this panel. I indeed am the Chief Medical and Scientific Officer at GametaCell, where I've been for 5 years now. The company has long been engaged in efforts to expand cord blood stem cells ex vivo and developed technology to do so using a unique nicotinamide-based expansion platform. And since then has developed a product candidate called Omidubicel, which is derived from a single unit of cord and expanded to increase the number of stem cells, and has been studied through phase 1, phase 1, 2, and phase 3, most recently in a randomized study versus unmanipulated cord, and has been found to increase the outcomes, improved outcomes for patients in terms of shortened time to engraftment, as well as decreased infections and shorter time in the hospital. And so Omidubicel is the lead product candidate that's being submitted for FDA approval. And GametaCell is also involved in development of other cellular therapies for patients with cancer and other serious diseases. My background is as a hematologist and oncologist, and I'm very passionate about working to develop novel therapies for patients.
Great. Thank you all. So you can see that we have a large view of cord blood and birthing tissues, both as therapies in standard transplantation, as source material or starting material for other types of cell and regenerative therapies. Some of us have experience in manufacturing, and some of us have experience in clinical trials, and all of us, I think, are navigating how to bring these products to patients, and what's the best way to navigate some of the regulatory pathways we all have been challenged with. I'm just going to mention that for those of you in the audience who don't know, cord blood is the only hematopoietic stem cell source for transplantation that is regulated, in our case by the US FDA, but also in other countries. And that's probably because it's allogeneic, but also cryopreserved and stored. It makes it different from other products, which are typically used fresh, like bone marrow or mobilized blood, although they can be frozen for later use. Cord blood also, when it's collected, is not directed to a specific recipient. It's collected, it's qualified, tested, banked, passes all the rules for donor screening and cell manufacturing in GMP, and then is available to be matched to a patient who's in need of a donor and doesn't have a donor in their family. Cord blood also is more tolerant of another individual. It's probably something in a simplistic sense left over from pregnancy, where the mother who's pregnant tolerates the baby who is made of half of the genetic material of the father. And yet in a healthy pregnancy, there's no rejection of that mother of her baby. And that property of tolerance is carried over into the cord blood cells. Functionally, that means that cord blood doesn't have to match as closely as some other donor sources and can yield similar results to matched bone marrow or matched mobilized blood in the unrelated setting. Even though we all have seen some of the dramatic results of cord blood transplantation. Marcy alluded to her contact with long-term patients, and I think I'm the oldest person in the room, but I have many patients now who I transplanted when they were babies or young children, who are young adults now and healthy and getting married and leaving home and going to college and having jobs. And I kind of feel like they're my children too. But they wouldn't be here if they didn't have a cord blood donor. And all of them have the story of being told there was no hope and there was no options, and go home and be comfortable, until you die. So we've all seen the benefits of cord blood transplantation. Having said that, cord blood as a donor source for transplantation is not being used as frequently as it was, say, 5 to 10 years ago. There are probably many reasons for that. Some are just logistics. Probably the biggest challenge to cord blood as a donor source is haploidentical transplant, using a half-matched family member, which has emerged as, at least on the front end of transplant, a simpler, maybe cheaper alternative to finding a donor for someone who doesn't have a full match in their family or in the registry. The thing that has really enabled haploidentical transplant to thrive is use of post-transplant cyclophosphamide as GVHD prophylaxis. Personally, and maybe Ronit will address this, I think maybe Omidubicel would enable that same approach to be used in cord blood someday. But certainly currently, you can find a haploidentical donor in a family, usually quickly, and harvest that donor without having to pay attention, frankly, to FDA regulations other than donor screening. That's really modified the willingness of transplanters to sometimes go to cord blood as their go-to source. Having said that, those of us who study cord blood transplant recipients have seen that long-term outcomes, so GVHD-free, relapse-free survival 5 to 10 years out, may be better with a cord blood donor, but we have yet to convince the community of that. So I just go around the room and kind of challenge each of you to think of ways we might be able to better message things about outcomes of cord blood transplant, and perhaps convince transplanters, if you think it's possible, to use more cord blood donors in the future. So Colleen, I'll start with you.
Okay, this is a great question, and hopefully this is going to incite some great discussion among at least the group here. And, I know this is recorded, so we can't get the audience involved, but I firmly believe that we need to stop some of the discussion around that one donor source is absolutely better than another donor source. I think there is now, a litany of literature, peer-reviewed literature, that shows not only is cord blood equivalent to matched unrelated and even sometimes match related, and I'll go into that a little bit more, but unrelated donors and haplos are equivalent, and haplos and cords are equivalent, and it's a bit of a different set of hurdles we go through to do each of these kinds of transplants. And really, I think the message needs to be that there is a donor for everyone who needs a transplant, and that really the transplant physicians in the field need to look at this holistically and look at what disease their patient has, because that may drive donor selection. Or, so do they have a non-malignant disease? Do they have a malignant disease? Do they have minimal residual disease, as Joanne had alluded to earlier? I think there is incredibly good data from multiple groups now, including ours at the Fred Hutch, that cord blood is a better source of cells to use for patients who do have residual disease at the time of transplant, because the relapse rates are lower. And that then goes back to the idea that with cord blood donors, unlike any other donors, we can actually permissively mismatch, meaning even if I had a donor that was absolutely fully matched at to however many loci you want to look at, say a 10 out of a 10 match, for a patient who has very, very high risk disease and who is at very high risk of relapse post-transplant, as a physician, you may say to yourself, well, maybe they have a SIB, but maybe that's not the best choice for this patient. Maybe I want to do a cord where I can permissively select a mismatch without increasing the risk of GVHD, but allowing that patient to have a better overall outcome. And I think it all ties together and goes back to that narrative of convincing people that cord blood can be done. It's easy to do. We can find donors very quickly. And just because you can skirt some of, as you know, the FDA regulations around the donor source, it doesn't mean that doing a haplo is the best thing to do. And so I think that's one of the things I'm very passionate about, is making sure we all talk about what is the right donor for my patient, and then choosing that donor based on that. The other thing I think that is not talked about enough is that we are a country, we are a world of increasing diversity of individuals that are being born. And those individuals, as they grow up, and some of them get cancer and some of them may need a stem cell transplant, we are not going to be able to find donors for them in the conventional sense that we do now. And if we do not maintain cord blood collection, these people will likely not have donors. And if they don't have a SIB or a haplo or someone else, someone in their family that is a suitably matched donor, they're not going to be able to move forward with their life-saving, their potential for life-saving therapy. So I think we need to think about this now, but we also need to think about this as what's going to happen in the future. And I think the increasing genetic diversity within our populations is a huge driver to maintain cord blood collection and to maintain cord blood transplant. Let's see. I think I'll stop there because I'm sure you all have more things to say, and we'll all pipe in after we all give at least an initial answer. Thanks.
Thank you. Those are all great points. Ronit, next. You're up.
Yeah, I echo everything that Colleen said, especially about the growing ethnic and racial diversity of the United States, which was documented in the last census. I think it's a national imperative to increase the cord blood supply in order to be able to accommodate our especially young population, who's extremely diverse. And what I'd like to kind of spend a minute on is thinking about what some of the barriers are to cord transplant in a world where other transplant sources that are less well regulated are being used more and more often. Sometimes experience by one or two anecdotal experiences can really color transplanters' attitude. And there is a smaller stem cell dose in a cord is smaller. And if they had an experience where a patient had a very prolonged time to engraftment, or maybe had a bad outcome because of the delayed engraftment, infection, or even mortality, that can color the experience of the transplanter and their staff. And you'll get fewer trainees and staff that learn about cord and want to do cord. And then there's less experience in the institution. And then once an institution doesn't do it anymore, or has done maybe one in the past year, there's a really high hurdle to getting them to do it again. So echoing what Colleen said, I think raising awareness of all transplant options and having the ability to offer a wider variety of transplant options to optimize the outcomes for patients, I think is a way to be able to serve the patients that we have. Now, certainly, from where I'm coming from in terms of technology, the development of technologies such as omidubicel could also potentially increase the use of cord blood, especially if there's a good experience. If engraftment is faster, if the patient didn't get infected, or that patient left the hospital after a few weeks and did fine, maybe that'll lower the hurdle a bit as well, and make cord blood more palatable to a broader variety of physicians. But I think there is an issue in terms of trainees and experience, which has been lost over the past few years. So an educational component is going to need to be important, and it's going to require collaboration among multiple stakeholders, including sort of a pre-competitive collaboration, maybe across industry, across blood banks, and across institutions.
Thank you. And Marcy.
Yeah, so Renee, I think that your point is really well taken. I can see it from the outgoing piece, right? So we see from our cord blood bank that where the units go for transplant is really regionalized, right? They go to 3 or 4 different transplant centers in the country, and then maybe we have one-offs to different places. So I think about why is it that other centers are not using cord blood from our perspective, right? And I do think that there is a lack of training, and it's really regionalized. The use is regionalized right now. From a cord blood bank perspective, we do a lot of work in our bank to have donors from across the country. So we're located in Cleveland, Ohio. We have collection sites here. We have collection sites in Atlanta and collection sites in California, with the intent that we can collect across the country. And it's a nice snapshot of the ethnicity that's today, right? And we know that that's changing rapidly before our eyes. And we really want to make sure that as a bank, we have units that are going to be there for everyone. I also am worried about the fact that families are getting smaller. So I would imagine, I'm not a clinician, but I would think that using a haplodonor from a parent to a child, their older stem cells, they're not going to be as effective as a sibling. And I think that the odds of having siblings, multiple siblings in the future is probably going to be less and less. So from a banking perspective, we try to be really thoughtful about our inventory and the units we bank and the sizes of the units we bank and the ethnicity so that we have one for everyone there. And I think the thing that keeps me up at night as a banker is when I hear stories of people that look for a match and they don't have one, because I also, I think there is a match for everyone if you include cord blood. So it keeps me up at night when I hear stories about people that can't find a match, because I think that that's not true anymore if you really do use all of the different sources that are available.
Great. Well, those of us who are bankers, but also Ronita's and Kalina's manufacturers, are keenly aware of the fact that FDA or similar agencies to FDA in other countries regulate cord blood, and in the US at least, the FDA strongly encourages licensure. They haven't mandated it, but the truth is that originally 7 banks in the US achieved or applied for and were granted a BLA. There have been some mergers and acquisitions. So now there are 6 banks that are actively practicing under a license and 1 additional bank that's just applying for a BLA. But the numbers overall are, in single digits. Having been someone who's lived through the BLA process and who at the beginning was not sure it really was valuable for banks. And I do think it hurt banks in the competitive sense of having a product that was just more expensive because of the licensure. But I also think there are some good things about licensure, about standardization of manufacturing, and also about enabling cord blood and birthing tissues now to be quote unquote GMP and potentially usable as starting material for other manufacturing. So what do you all think about licensure? At the end of the day, do you think overall it was a positive thing or a negative thing? And Marcy, I'll start with you.
Sure. So on the onset, it was really, really challenging, right? It was expensive for us. It was, we were kind of backing into it. I think when you go into making a product that you're going to do a BLA, you acquire those skills along the way when you're doing that. So we were already releasing products. We were already had established procedures, and then we had to back into a BLA. So that being said, it was challenging. It was also a new mindset for a lot of our staff, right? To have a major change like that in the middle of already being proud of the work that you do. And now this agency is coming in and saying that what you were doing wasn't good enough. So I think that took a lot of work to say that this was going to make us better in the long run. But what it did do that we didn't anticipate was it really made us standardize our procedures. It made us do a much better job of our document control and our error management, which actually made us extremely more efficient in the long run. Although we had a lot more complex things to do, we had no choice but to become more efficient about them. And there was no wavering on how we were going to do things. So we got our license in 2016. It took us a long time to get it, but I think the benefits going forward were actually excellent because we knew exactly how to do things. We have learned how to do things and change them with the FDA. And so I think although it might stifle innovation a bit because it is challenging to change anything, we've also learned how to navigate that. And then I think in the bigger picture, now that we're talking about using cord blood as starting material, about trying to develop our own uses of cord blood outside of transplant in our research and development laboratory, we know exactly what we need to do to follow that new path to an IND. We were already doing it with one product. So translating that to a second or third or fourth, or partnering with people to help them to do that is really simple for us. Our staff has grown in their capabilities through it, and we've been able to actually keep most of our staff through it and grow them, which I think has been a real growth opportunity for them too. In this era of really challenging staffing, we have maintained our staff, and I think it's value add for them to get new skills too. So I think in the long run, it was actually profoundly a benefit, but it was really challenging in the beginning.
Thank you. So Renita, in Gamida-cel's experience at manufacturing Omidubicel, what has been your approach to requiring units that you manufacture to come from licensed banks?
Yeah.
And maybe you could also address, the issue of HLA diversity and the fact that there's a big inventory of units collected under IND that may be good matches for patients who would get a product manufactured or expanded through your process.
Yeah, absolutely. Well, first of all, the cord blood banks are a critical partner for us, for Gamida-cel in particular, and just for industry in general. And we've had very productive collaborations over the years with a number of cord blood banks. One of the challenges working with multiple cord blood banks to develop the product, as well as to create and manufacture the product for many patients, is the consistency between banks, and it's a very hands-on process to get information from banks, to fill in missing data, to work on the logistics, and to interpret some of the idiosyncrasies. So having the consistency and standardization that the licensure affords is extremely important. In a clinical trial setting where we were able to use all sorts of cords in order to meet the needs of the patients under an IND. In the commercial setting, we may be more limited in the types of cords that we'll be able to use in order to keep to the standards. But the most important piece is really the work with the cord blood banks. We have a very hands-on process to work with cord blood banks and get the information, as well as get all of the shipping and logistics and all of that done. In terms of HLA, rare HLA types, I think it's important to think about being able to collect, identify, and have in the inventory the rare HLA types, and if those could be licensed, I think that's a benefit for all.
And Colleen, as a manufacturer, how do you feel about starting with material from a licensed bank?
Well, we obviously, like that. And I think we're going to talk a little bit more about the cellular starting material piece too. So I'm going to hold some of my comments on that, because I do think it's actually really important. And it's important when you're writing a new IND on products that are not standard cord blood for stem cell transplantation. But I think the whole issue of licensure and going through that, developing an expansion technology that was already in use clinically, when the legislation started to hit and this requirement for the banks to become licensed, I have to say, even from a clinical standpoint, it created a lot of confusion, and I was really concerned when this started happening, because we had a very vibrant cord blood program that was growing based on the research that we were doing. And all of a sudden it was like, wait a minute, if I want to get a cord blood unit from a bank, I need to do it how? And it's under an IND, and it's a blanket IND. And what does that mean? But is it licensed? Is it not? What's the cost going to be? I think there was so much concern about that. And I think there still is confusion today. So as a manufacturer, there's still a lot of confusion when I talk to, say, investors or other people about cord blood and how do we get it and how much does it cost, and is it the same cost as, you know, getting a unit for stem cell transplant, and you know, the fact that all the banks had to name their product was also very confusing for everyone, right? And I think even just recently, I think someone sent me, I can't even remember what the story was, but it was about, and it used the product name, and it made it sound like it was this more than minimally manipulated cell therapy. And so I think there's still a lot of confusion unless you sort of are right in the middle of it and you either lived through it. And, you know, in the end, once people sort of took a deep breath, I think the clinical centers, the transplant centers, once they sort of figured out, okay, well, I can still get this, I just need to get it under this blanket IND, or I need to get a licensed unit, it was all fine. But I don't think the FDA really anticipated how much confusion would it do from doing what they did. And I think this goes back to our previous conversation about how do we ensure that cord blood is still utilized? How do we ensure that people are still being trained? I think I can say this because I'm a transplant physician, but transplant centers tend to pick a source, right? We don't tend to see, that's why, Marcy, you're only seeing your units going to 3 or 4, the majority 3 or 4 centers, is because people tend to pick, I'm going to be a haplo center, or I'm going to be a cord blood center. And I think we just need to change that narrative as well, because otherwise, as Ronit said, we're not going to have fellows who are trained, and then they're going to have that bad experience. So if you don't do a lot of cord blood transplants, and Marcy, you got that patient who's like, I can't find a donor, that kills me too. It kills me every time I read a story about that, you know, about all this work to find a donor when I'm like, you've got one sitting in the freezer, most likely. So that sort of kills me as well. But I think, you know, wait too long, then you have a patient who's been very, very heavily pretreated. They should have been transplanted months ago. They come to transplant, they don't do well, and they're like, see, doesn't work. So I think it does require collaborative effort. It does require patients who, so if there are any patients listening to this who have had a cord blood transplant a long time ago, you know, telling your story, saying what it was like for you, saying that, you know, what your life is like now, I think are all very important things. And the licensure part, I think, does help, you know, in the end. I think the knowledge that there is consistency within, you know, the bank and between banks because of the regulations is an important part of it. And it certainly makes it easier as a manufacturer to use that material as material.
Great. Yeah, I mean, I will say, I'm going full circle back to me, but as a banker, when we first faced the challenges of getting the BLA, which for us really revolved around more control of our facility and more control of our documentation, it felt hard. It felt not necessarily necessary. But now that I look back on it and I see what we've put together and how we control things moving forward, I do think it adds value. I will say though that the engraftment and you know, complication rate, if you want to call it that, with our IND units versus our units collected under our BLA are not different. But I think the quality of the activities that go on in our bank is much higher under the.
Yeah, and I think that's super important, right? Because you think about all the units that have been collected that are accessed to an IND, the outcomes probably are this, right? The material, like, so you're saying your material, you don't feel like your material changed so much, but I guess moving forward, I imagine that having these kinds of the document control and all these other controls and facility controls, I imagine that translates overall to more consistency and overall better units, and being able to look back on your inventory and be able to choose how you move forward, what donors you process, or whatever the case may be. So
I often describe it as the fact that the bag itself is not any different, nor are all the things that came with it, but in the old units or the IND units, in order to find all of the things that belong to that unit, every person that touched it, every piece of plasticware that we use, what the temperature was, what box it came in, we could have found it, but it would take a long time. Now with the licensed units and our document control and our EM monitoring, we can find all of that immediately. So it streamlined all of it. It was all there, but it wasn't very well organized, at least in our shop.
No,
And that's exactly our experience too, from a manufacturing perspective. It's really about the information. It's about the detailed unit reports. It's about handling of missing information, how many times we have to go back and forth to get the missing pieces. So it's really around that. It's not the quality of the unit itself, the size, or anything, which are all separate and all relatively consistent, but it's about the pieces around it that the consistency makes it easier to execute. And that's very important too.
all right, well, we have roughly 15 to 16 minutes left for the rest of our conversation. And I wanted to take a few minutes just to talk about cord blood and birthing tissues in general as a starting material for manufacturing other cell products and regenerative medicine products. I think, Marcy, you were the one who once said to me, the worst thing you could do with cord blood is to throw it away, right? So, because there are so many options now for its use, whether it be in research or it be in manufacturing of other products. So I think, we want to make that point that we've put together these laboratories that are now licensed in GMP that have experience and know how to collect material in a GMP way, which is very important for starting material for manufacturing of these various products. And I think the cord blood banks don't really get enough credit for having weathered the journey to figure out how to take an IND process and get to a BLA and standardize that. But now that it's there, capturing not only the cord blood, but maybe the cord tissue, or maybe the placenta, or maybe the amniotic membranes, is expertise in biobanking, if you will, that cord blood banks now have. So I want to just spend a few minutes, shorter comments maybe from each of you, about what you think about whether cord blood banks are good vendors for manufacturers who need starting material, and whether or not you think that material should or should not be collected under GMP. Colleen, I'll let you go first.
Yeah, well, they're critical. I mean, they're absolutely essential part. So there is no way that I want to go out and try to recreate what the banks do, especially for folks like us who are developing allogeneic therapies, right? These are therapies that we need, qualified starting material to then make our products, which will also be put in a freezer and do not have a patient attached to them yet either, right? So these are not autologous or patient specific. I think, unequivocally, it is critical for us to have relationships with the cord blood banks. And I do believe that we should get the material from licensed facilities because of all the things we've already talked about. I do think we are in a critical phase of growth, especially of the allogeneic cell therapy space. And there is a lot of confusion over the cellular starting material and how that is regulated. And when does it go into the more than minimally manipulated? Because, I, as a manufacturer, we want to get product that is fully qualified, but that's our cellular starting material. And we rely on you to collect it as appropriate per the regulations, right? 21 CFR. So I just think that then we're going to take that more than minimally manipulated And how does the donor eligibility play for my starting material versus my final drug product? So I think there's a lot of work to be done right now around these issues and the confusion around these issues. So we're all speaking the same language, not only for you guys as the bankers, but from the manufacturer side of things.
So Marcy, what do you think about consenting moms through the cord blood bank for these additional uses of cord blood or the birthing materials?
So I'll tell you, in our experience, we have redesigned our consent form a few years ago, and we made it as broad and as transparent, talked about commercialization, talked about things that might be done with it. We were actually anticipating seeing a reduction in donations, to be frank, right? We thought that we would see people say, no, thank you, I don't want to do that. We were very clear about the fact that they would not receive any compensation for this donation, nor if we used a commercial partner, there would be no compensation back to donors. And, you know, we didn't see any pushback. We saw some longer conversations. We did a lot of work with our consenters, who are mostly nurses, to talk to them about what kind of questions they might get. We fielded some phone calls. We still get some emails about it. But I think that in general, donors understand it. They either want to really help move the cause, and we've really taken the stance in our bank about use for transplantation is one thing, and then use for commercialization, research, and we have an opt-in box. So you can say, I just want it to be used for transplant and nothing else, or we could use it for quality control, commercialization, research, anything. And I'll tell you that we haven't really seen a drop off in people that check that box. We wanted to be as broad as we could. And I think the donors want to see it be used for something. I think throwing it away or having it sit in a freezer for 10 years is not why they donate.
And Ronit, your thoughts on this?
You know, one thing that using cord blood for a number of different, as starting material for a number of different potential therapies will do is sort of raise the profile of cord blood, right? And that helps everyone because it raises the awareness around cord blood transplant and raises awareness around cord blood donation. And all of those things are critically important for all of us to succeed. So I think it's a wonderful starting material for a broad range of therapies, and getting that word out there that we're using it for all those potential therapies is really important. One thing that I think that the cord blood banks can do to partner well with industry around this would be the availability of custom inventory searches, looking for specific parameters from the starting materials to enable manufacturing of certain types of products. So I think that would be helpful. And again, the sort of the consistency and ease of use of documentation, all of those things are important to continue to improve upon as well.
Great. Well, let's end the session in the next 10 or 12 minutes by talking a little bit about what the vision of the future is and what these emerging uses are looking like. And I think each of us have a little bit of experience in those areas, either providing material or actually creating products. So I'll go first, but then have everyone talk. In our work at Duke, in addition to looking at cord blood and transplant, we started to look at cord blood as a cell therapy without transplant in kids with cerebral palsy, babies with birth asphyxia, adults with stroke, children with autism, and actually adults with COVID ARDS. We've run phase 1 and 2 clinical trials in those areas, initially using autologous cord blood to first show safety, then sibling cord blood that was haploidentical or fully matched, and then finally allogeneic unrelated donor publicly banked cord blood, which is where we've landed to take some of these studies into phase 3. And the reason we landed there is because of control of the product, and I think more importantly, the fact that we observed a dose effect and a dose threshold. And for reasons I still don't understand, the dose was 25 million cells per kilo, which is the same dose that's kind of the threshold for hematopoietic cell transplantation with an unmanipulated cord blood unit. We think in the work we're doing, the active cell is actually the monocyte, not a hematopoietic stem cell, and that the monocyte can signal through macrophages in the lung to then signal oligodendrocytes in the brain to proliferate and remyelinate damaged tissue. So it's not a stem cell effect. It's not an engraftment effect. It's something very different. It's, as Jan Nolte says, it's using the cell as an intelligent drug. We're exploring that again in kids with CP in particular. We're also manufacturing cord tissue mesenchymal stromal cells, third party. And the advantage, if you want to think of it that way, of these cells are you can make large batches from a single umbilical cord. So instead of what you experience with a cord blood unit, which is generally 1 unit is 1 dose. And if you need more, you have to expose that patient to multiple donors, which for other reasons may not be a good thing. Using cord tissue MSCs, you can get multiple doses for hundreds of doses out of a single cord. It's definitely a 351 product. It's more than minimally manipulated. It's complex manufacturing. But it's creating a product that can be used for many, many patients and can be given multiple times from the same donor. Now, the cord tissue MSCs regulate inflammation best that we can tell, and others have done a lot of work in this area, but they're not promoting remyelination or some of the things that the cord blood monocytes do. So I think there's roles depending on the disease you may be treating for both products. But I'll give one example where in HIE, which is hypoxic ischemic encephalopathy, this is newborn term babies who have a hypoxic hit right around birth and are born very depressed, have to be resuscitated, have a very high mortality, are cooled, which helps survival and probably brings survival to 80%, but at least half of those babies have severe cerebral palsy for the rest of their life. So we added a initially autologous cord blood infusion to that cooling therapy, and we saw benefit. But when we took it from phase 1 to phase 2, we ran into logistical issues because most of the OBs delivering the babies just couldn't or didn't have time, had to attend to the sick mom and the sick baby, so they couldn't get enough cord blood for it to be used therapeutically, even in a newborn. So we switched gears and went to the cord tissue MSCs, which are quote unquote off the shelf, don't have to be matched, don't engraft, and aren't a risk to a newborn. And we've seen some very encouraging results in phase 1, and we're going to take that on. So to Colleen's point at the beginning, when she was talking about cord blood donors and other donors, even with the cord and birthing tissue derived cells, there are going to be multiple kinds of cells, multiple kinds of therapies, multiple uses, and there's not going to be a one size fit all, in my view. So Colleen, why don't you kind of talk about immunotherapies and the work you're doing?
Sure, and I'll try to just keep this brief because I know we're running out of time, but I think this is such amazing. Okay, I think this is such an amazing time to be a part of this field. And, we really have, my group has taken the stance that we are going to be very focused on developing allogeneic off the shelf, meaning, products that can be given to anyone without the need for HLA matching, because we feel that that's going to be the fastest way to get therapies to patients. And there's going to be hurdles with that. But we've kind of come full circle. We started in this land of patient-specific expansion for cord blood transplant, specifically to help with engraftment. And that was beautiful, but we really decided pretty early on that we wanted to shift gears and make products that could be given by any physician, regardless of what they were. They could choose their own conditioning regimen, they could choose their own whatever and give these things. So in the end, we've kind of come full circle. We initially used our expanded stem and progenitor cell product. Again, it was off the shelf though, right? So these cells don't persist for sure, right? And that's one of the hurdles, is can they persist long enough? And we found that approach and the fact that cord blood transplant recipients just actually with better quality, higher quality, bigger doses and donors, the time to engraftment isn't as much of an issue anymore. And so we pivoted, and we're using the same product now as a non-engrafting cell therapy to help induce remissions in patients with acute myelogenous leukemia. So we have the beauty of this approach, though, is that we have all of this data from our historical studies that we can then apply to this product moving forward as an off-the-shelf. But the other immunotherapies that we're working on are all cord blood derived, and we always start from the cord blood stem cell. And so we are developing natural killer cells. And I have to be very clear, there are many people working with cord blood derived cellular therapies, immunotherapies, and NK cells are a big one in the field right now. And they can be engineered, so we can make CAR NKs. And the hope is that these types of approaches that are allogeneic and off the shelf can be dosed multiple times if they need to be, but can be all made ahead of time. So you don't have the logistical difficulties nor the expense that we see in the autologous CAR T space. So there's natural killer cells for malignancies. We're also using unmodified natural killer cells for malignant indications, AML in particular, as well as an antiviral approach also in COVID-19. So for patients who have been diagnosed and hospitalized, but not yet in the ICU, which is where we're seeing MSCs being used for patients with ARDS. And we're working on MACs. I guess that's the last thing we're doing here is cord blood derived macrophages. So we're generating them from stem cells. And this too is a product that can be used either as an unmanipulated or unengineered, non-engineered cell product or an engineered cell therapy. And the cool thing is we can start to think about combining these approaches. Can we combine our engineered MACs to help our NK cells or whatever the combination may be? But there are other companies, other groups, other academicians working on cord blood derived T regulatory cells for multiple indications, cord blood derived cytotoxic T lymphocytes as antivirals. So I think the future is bright. We need to make sure that we do continue to collect cord blood and that the banks survive all of this. So any women out there who are listening to this, who are pregnant or going to be pregnant, think about this, right? You can really help the field here. Yeah, and I'll stop there because I'm sure Ronit have lots to say too.
Okay, Ronit, your thoughts?
Yeah, it is indeed a really fantastic time to be involved in cell therapies. There are hundreds of cell therapies in development, and there's a lot of excitement and anticipation about the way that they're proceeding in the clinic through clinical trials and hopefully more and more towards approval. From my perspective at Gamida Cell, we're looking forward to working with FDA to move omidubicel to approval in patients with for stem cell transplant in patients with hematologic malignancies. And beyond that, developing off-the-shelf or readily available therapies in an allogeneic fashion to treat different diseases as a cellular immunotherapy is a goal of Gamida Cell as well as many other companies. So indeed, a lot of bright things in our future.
So Marcy, other than me saying goodbye, you're going to get the last word, and I'll start you off with younger is better.
Right. So I think from a banking perspective, we only bank a small portion of the units we use. And we used to, if you think of the clinical development pathway from R&D all the way through to a BLA. Now we have a BLA and our frozen units get sent out for a BLA. We typically sent fresh units to research and development, academic users that were close by. But now we're seeing users all across that continuum in phase 1, in IND development, in phase 2. So I think I would be worried if we didn't see those research unit users for these new indications. We've worked really hard to increase our utilization. We've had days, our lab celebrates when we have 100%, where every unit that comes in finds a place to be used. And those are big days around here. So I look forward to those days becoming more and more frequent.
Great. Well, I want to thank all of you for this hour of conversation. I hope everyone listening benefits from our insights, and we'll close just by saying goodbye and thank you all.
Thank you.
Thank you.
Bye.